Thank you for joining our webinar, Aggregating and Sharing Clinical Evidence to Improve the Diagnosis of Rare Diseases. My name is Ben Liesfeld from Limbus Medical Technologies. I'm going to talk today about how to apply whole exome sequencing in routine diagnostics. By the way, if you have any questions during the talk, feel free to ask the questions in the chat. I will be happy to follow up with them via email after the webinar. I will touch 4 topics during my talk. First, I'm going to explain a little what is holding us back actually. Whole exome sequencing has been around for a few years now, but it's still not widely used, and it's certainly not used as a first-line diagnostic. Why is that? We'll look at this in more detail, and we will highlight the critical steps in the whole exome sequencing process, and I will explain what the potential pitfalls are, and then I will show you a validated solution that enables you to actually achieve an optimum process in diagnostics. And I will explain with a few clinical examples what the specific benefits of a validated solution for this process are. So first of all, why is whole exome sequencing not performed as first-line diagnostic everywhere? There are a few issues that are most frequently raised. So the first one is typically whole exome sequencing is too expensive. It's not worth the money compared to targeted sequencing. The second issue is that review is too difficult. People don't feel prepared to do the job. It's very time consuming, and there are typically too many variants to review. And third, this all causes the turnaround time to be very long. It takes a long time to return results to the patient, and many of those results are inconclusive, so you cannot even give the patient an answer. And this is something that not only small laboratories who are just starting are struggling with, it's also large and very experienced institutions who are struggling to achieve a high diagnostic yield from whole exome sequencing. Let's take a look at the whole exome sequencing process in more detail. This is a very crude depiction of the process, and the steps are somewhat arbitrary. But on the left-hand side, you can see the wet lab process, starting from DNA extraction, library preparations, actual sequencing, and on the right-hand side, it's rather the software processing, the data processing, and the interpretation steps, the bioinformatics, filtering, and the reporting. And so we have identified in our work with our customers that, well, since the sequencing instruments have matured so far, they have grown to a large capacity and they are generating data in large batches, and you're then struggling to actually process all this data in the next step. And you're most of the time even getting slower when trying to interpret these large amounts of variants. So what are the critical steps, and where do you have to pay attention in order to streamline the process and to maintain high throughput through all the steps? So actually, one of the wet lab steps we identified as very critical in whole exome sequencing, and that's the library preparation. And the issues that you may face there are you may have a GC bias, and you may not even be aware that there is a GC bias, or there may be other reasons why the coverage in your targeted sequencing is non-uniform. Or you may have great variability across samples or across batches of samples. And all these issues impact how the downstream processing and the interpretation can go on. And so you may have impact on the diagnostic yield because you have gaps in coverage. And some of those issues may impact the sensitivity in the CNV analysis. And in total, the whole process, the entire assay will not be cost efficient because you will have to compensate for those issues in the library preparation. So it really makes sense to address those, to identify those issues in the first place, and to address this in the very first step. Another critical step in the process is certainly the bioinformatics. And what we've seen in the field so far are setups that are incapable of reliably calling CNVs in the data. Software setups that are not standardized, that are not even validated, that require a number of manual steps. And those setups are certainly not scalable to a degree that is required in routine diagnostics. And this impacts, of course, the diagnostic yield. If you cannot even see copy number variations, you cannot report on them, and they may make up a significant fraction of your cases. You will have variability in the quality and in the results of your diagnosis. You will have prolonged processing time. You will have a bottleneck here. And in general, you will push workload from the bioinformatics into the interpretation part. So, for example, the reporting team will spend a lot of time figuring out if a variant is actually there. So they have to do a lot of quality control, and this is the wrong place to do that. This just creates workload where it shouldn't be. And the third critical step that we mostly address is the filtering and the classification of variants. And the issues that you may be facing there is that you have too many variants that you are supposed to review, that information that you need and that you're utilizing is distributed across many tools, and that you have too many inconclusive results. And the reason for that is probably that you have not sufficient reference data to be able to compare the variants in your patient to. And this will impact your ability to correctly interpret the significance of variants. This will impact your ability to actually perform diagnosis on your patient. And it will impact the turnaround time. It will extend the turnaround time to a long period of time. And this means that costs are increasing. This part actually in the workflow involves the most expensive labor there is in the laboratory, and you want to structure this as efficiently as possible. So overall, these issues may impact your ability to apply whole exome sequencing in routine diagnostics in general. I stressed the term reference data and the ability to use it in the previous slide. And so what is reference data actually, and what can it be used for? So the first thing that comes into mind when talking about reference data in genomics is, of course, databases, collections of known variants that have been reported before. Of course, one of those databases would be ClinVar, or you could also consult Mastermind, which is a very comprehensive database of published literature. And if you're looking for copy number variations, you would probably consult Decipher. Another source of information, which is reference data, is information about known phenotype relationships. So relationships between genes and phenotypes that have been described before, and you could find those information in the human phenotype ontology or in the OMIM database. And the third kind of reference data is generated from healthy reference populations. One example is the GNOMAD database or the GGB database if you're thinking about copy number variations. And last but not least, there is a source of information that we call real-world evidence. It's called that because it's not being generated or has not been generated during controlled clinical studies, but it's continuously generated during routine diagnostics. So it's made up of data created from real patients. It could be that this kind of data is located in proprietary in-house databases. So there are some large institutions who have the manpower and the funding to maintain and to create those proprietary databases. But you may also find real-world evidence in aggregated databases where several institutions contribute and share information. So how can these types of reference data be used when diagnosing patients, when analyzing whole exomes? So there are different purposes of these data types. So first of all, the known variants you can use to reduce the number of false negatives. They help you avoid to miss a variant that has been previously reported. And in the same way, the phenotype information works. It helps you to prioritize, to rank the genes to the degree that they overlap with the known phenotype of the patient. So they help you to reduce numbers of false negatives. And then on the other hand, the reference data from healthy populations work exactly the opposite way. They help you to filter out all variants that just appear too frequently across healthy populations. And therefore, the simple argument is what is occurring too frequently cannot be pathogenic. This is a thinking that is valid in this setting of dealing with single gene diseases, and where the explanation is a single variant. And last but not least, the real world evidence gives you both. It gives you the ability to reduce false positives, like the healthy reference populations, because every patient that is contained in this real world evidence database can serve as a control for the patient that you currently look at, because pathogenic variants are just so rare. And the real world evidence can also serve to reduce the number of false negatives, of course, because you have access to additional evidence to be able to classify variants as pathogenic. I'd like to explain a little bit more what the quality of the data is that is contained in real world evidence databases. If you compare, or if you're looking in more detail at GNOMAD, GNOMAD is a database that has been built from data obtained from healthy adults. It has been curated very diligently, but still you may know that there will be some pathogenic variants in this database, just because there may be variants that don't have a high penetrance, or variants that are just related to a specific risk to have a disease. And if you compare this with the variants that are contained in real-world evidence databases, you will see that while those databases will certainly contain a large number of frequently occurring variants that should be considered benign, but of course they also contain a larger fraction of pathogenic variants, and very importantly, they contain NGS-specific artifacts or variants that are frequent because they occur in low complexity regions. And those last two types of variants, they are specifically excluded from the GNOMAD database, for example. So you will never find them there, but you will find them in your patients, in your real-world data. And this means that being able to compare to a real-world evidence database will help you better classify your variants. If you're using such a database, make sure that you also have access to technical metadata, and of course also to phenotype data. It's very important that if you're comparing to other patients, that you know what these patients look like, and if these phenotypes have anything to do with the phenotype of your own patient. And those databases should also contain classifications of variants and the final diagnosis of those patients. Please always keep in mind that if you're talking about and if you're dealing with patient data, since genomics is such a new field, the information will change. So it's very important to have mechanisms in place that can deal with updates. Data will be updated continuously. And of course, there will also be conflicts. So you have to have mechanisms that deal with conflict resolution. If you don't have that, we have a good example what happens then in ClinVar. You have conflicting assessments of variants, and they make it very difficult to deal with this information and to really make use of the data that is contained in ClinVar. So now that I've talked a lot about the theory of data sources and how you can utilize them in the whole exome diagnostic process, I would like to explain how a good solution, a validated solution, can look like. And this is our software solution, VARVIS. So VARVIS covers basically and supports the workflow starting from the raw data from the sequencing instruments. It covers the 3 steps on the right, the bioinformatics, the filtering, and the reporting. But most importantly, in the very beginning, it gives you immediate feedback about the quality of your data. And this is something that we work with our customers very closely is monitoring this the laboratory process and helping them to optimize and to achieve the level of quality that's required for routine diagnostics. The software presents to you a very clean interface to collect all this data. All the data is going and stored into a database, so you have the ability to monitor data over time, and you have the ability, you can set thresholds for quality parameters, and you will be notified if any of your samples fails any of those thresholds. So you have a very quick overview if everything is going to plan, and you also know very, very quickly if you have to optimize into a certain direction. Of course, a core component of VARVIS is the bioinformatics component. And since VARVIS is a cloud-native decision support software based and operated in the cloud, it is very easy to set up, and you have no investment to make into IT infrastructure. That means you also can scale up very quickly from small panels to whole genomes, or from a small number of samples to a very large number of samples. But what is very important from our point of view is that the processes are validated, and they are validated both in the SNV analysis and the CNV analysis. And we actually provide this validation as a service to our customers. And this is how this looks like in practice. Our customers simply sequence specific reference samples as per our instructions, and those samples go through the regular process as any other sample in your laboratory. And we then compare the results in an automated fashion with the reference data. That means you obtain automated validation reports every time you sequence such a sample. And we also repeat this validation every time something changes in the software. So you can always be sure that the results that you obtain from the software are valid. The same level of diligence we also apply to the CNV analysis. We have performed validation projects on thousands of samples obtained from our customers. And we always compare it to the gold standard in CNV analysis, which are PCR-based methods. And we obtain 100% sensitivity if the samples comply with our quality criteria. And at the same time, we achieve a very high specificity. And this is very important if you want to apply this technology as a screening tool, as first-line diagnostics. If you have a very high false positive rate here, this would be very cost inefficient, because you would have to perform a lot of additional testing to confirm the many variants that you find here. So in practice, our customers have all samples being analyzed for CNVs. There's a specific quality control because the CNV analysis is more sensitive to quality than the SNV analysis. But in the end, most of the samples are undergoing this process. And since we achieve such a high sensitivity, you can be sure if you don't find a CNV in the data, you can be sure that there is none, and you can very quickly write a negative report. So then the other step in the process that we pay a lot of attention to is the filtering and the classification. And I explained before what the real world evidence is and what it is made up of. And we enable you to capture this evidence and capture the entire picture of the patient. So it starts from data about how the data was generated, how the wet lab process was performed, information from the sequencing instrument, information from the bioinformatics processing, and you can amend this information with family relations, with the entire family history, and of course you can add the phenotype, the classification of variants, and the final diagnosis. So you bring everything together that you have about a patient, and this is stored in a centralized database. And this then enables you to actually systematically manage the data that you are generating. So in the beginning, we enable you to capture the data from the single patient using standardized ontologies. But then you are collecting with every other patient that you're diagnosing, you're building your own local database. And this database typically consists of homogeneous data sets, and you have full access to the analysis history. And on top of that, we aggregate data from many different institutions and create a reference data network. And this allows you to have access to many data sets in compliance with international regulations. And I'd like to explain in detail how this works and why we are compliant with international regulations. The protection of patient data is crucial in the setup and is implemented into the software by design. So if you think of two laboratories, laboratory X and Y, who are seeing different patients and who are performing diagnosis on different patients, they transmit only de-identified data to a centralized database. We're only interested in phenotype information and in genotype information. And if you now think of yourself or your laboratory performing whole exome sequencing on a patient, and this is maybe the first data set that you're seeing, you receive annotations to every single variant in your data set from the centralized reference data set. So you receive information, how often has this variant been found in other patients? Has this variant ever been classified in other patients? And if it has been found in other patients, what are the phenotype descriptions? What are the HBO terms that are associated to those patients? But it will never be possible to you to actually access the entire data set of a different patient. This is entirely protected. Now some facts and figures. What is actually now contained in the database? This is data from March of this year. We have information in the database of more than 19 million unique small variants. The data has been generated from 35,000 patients. Those patients are part of 21,000 different families, and 17,000 of those patients have been described diligently using standardized ontologies, which makes the data very useful in the analysis of whole exome datasets. We also collect information of copy number variations. So at this moment, more than 100,000 copy number variations are contained in the database, and this is data that was generated from more than 6,000 different cases. So this is already a very powerful and very comprehensive collection of datasets. And now I'd like to use a few examples to highlight the utility and practice of those datasets. So my first example will show in a very simple manner how utilizing the ALLEXES database can accelerate your search for significant variants. So in this example, this is a diseased child. The parents are healthy, and the entire trio was sequenced using whole exome sequencing. We entered the phenotype into the software, and we are also entering the family relations between the individuals into the database. So all three data sets are linked and can be analyzed accordingly. This is being utilized, this information is being utilized by the software in the analysis. So let's go through the data review step by step. We are basically starting with more than 40,000 variants that have been found in the patient. I'm now applying very simple filtering against the GNOMAD database. This is a very severe phenotype, and I'm filtering only for very rare variants in the GNOMAD database. So this reduces the set that I'm looking at to a little more than 1,000 variants. If I additionally apply a filter against the ALLEXES database, I can even further reduce this number by a factor of 2. So it takes away 50% of the variants that I would have otherwise further downstream have to look at. So this is a very powerful filtering, and it works in the same way as if you already had sequenced tens of thousands of whole exomes yourself. And the filtering works very easily in the software. You can store any filter configuration for your data sets in buttons, and you can see here in the screenshot, the yellow buttons are the activated filters, and just with the press of 2 buttons, I can get to the stage of filtering. I now apply further filtering for impact of those variants with respect to any known transcript of the genes that they are related to. And I can also apply a filtering, or rather a ranking, of my variants with respect to how well the phenotype of the patient matches with known phenotype descriptions of the genes. So I end up with about 30 variants in the end that I would have to look at in more detail, but I haven't made use of the family relations yet. So I'm now filtering for different hypotheses what the inheritance pattern would be in my trio data set. And I then end up with just 3 variants that would fulfill an autosomal dominant inheritance patterns, and 3 variants from autosomal recessive compound heterozygous inheritance pattern. So in the end, I only have to look at 6 variants in detail, and this is a manageable number. So it's always the goal of the reviewer to reduce the number of variants to look at below 10. Anything above that would be too much to handle and too much work in a routine diagnostic process. So this is a screenshot of the result in the software. You can see that the filters that I mentioned were applied. These are, in the screenshot in the back, you can see three variants that are left in the autosomal recessive compound heterozygous filter. And one of those variants is selected. The result is that 2 of those heterozygous variants are identified as causative for the phenotype in a compound heterozygous state. And you can also see that these variants are now known in the ALLEXES database, and you wouldn't even miss them because you can now filter also for pathogenic variants in those genes. My second example highlights how important it is to perform CNV analysis on all of your samples. The CNV data from whole exome sequencing is so rich, it can basically replace array diagnostics. And this is an example where 2 de novo CNVs were found. And this is so simple that it can be seen at a glance from those data sets. This is a screenshot from the exome trio. So you can see at the top the index patient, the child, and at the bottom the father and the mother. And there are 2 variants visible. One variant, and both variants are de novo because they are not visible in the data sets of the parents. So there's 1 deletion on chromosome 21, and nearby there's also a duplication. So this was, again, a very clear case and very easily visible, and highlights very nicely how much the CNV analysis complements the SNV analysis in the clinical diagnostic. So this is a slide just highlighting what you can do if you have access to an optimized software platform in using whole exome diagnostics. There's an example of a laboratory that started working with us in 2016. In 2016, they didn't do any significant number of whole exomes yet, but as you can see, by utilizing the power of the VARVIS solution, they were able to basically double the number of samples that they're processing every year. And from the numbers of just of the first quarter, they are going to process a few thousand samples in this year. And I have to point out that they don't have a bioinformatician that is involved in the clinical diagnostics process. And they have published, as you can see here in this link, that a trained scientist can easily handle several hundred cases per year using this kind of setup. So this concludes my presentation how a diagnostic process for whole exome sequencing should be set up. We are very frequently discussing topics around whole exome sequencing in our blog. Feel free to check it out and to provide us with feedback if there are some topics that we haven't touched so far. Maybe you're also interested in workshops about clinical diagnostics using whole exome sequencing. We are hosting these kinds of workshops, and the workshops are conducted by clinical experts with a lot of experience with whole exome sequencing. If you're interested, get in touch with us at events@varvis.com. Thank you.